Cell biology is an intriguing discipline that examines cell structure, function, and behavior. Knowledge of cellular processes is critical in disease diagnosis, treatment, and medical research progress. Gel electrophoresis, a key technique, separates and analyzes molecules based on their physical properties. We will explore various gel electrophoresis techniques, their applications, and their importance in cell biology research. Join us on this exciting exploration of gel electrophoresis. Polyacrylamide gel electrophoresis, PAGE, is a technique used to separate proteins based on their size and charge. A gel matrix is created by polymerizing acrylamide and bisacrylamide molecules, forming a porous network that traps proteins as they migrate through the gel. By comparing the migration distances of proteins to molecular weight markers, unknown protein sizes can be estimated. PAGE is useful in protein purification, characterization, and identification studies. Agarose gel electrophoresis is a technique used to separate DNA fragments based on their size. Agarose, a natural polysaccharide extracted from seaweed, is used to create the gel matrix. The gel is poured into a horizontal or vertical electrophoresis apparatus, and DNA samples are loaded into wells at one end of the gel. When an electric field is applied, DNA molecules migrate through the gel, with smaller fragments moving faster than larger fragments. After electrophoresis, the DNA bands can be visualized using DNA-specific stains, allowing researchers to analyze DNA samples for purity, size, and quantity. SDS PAGE separates proteins by their size using a detergent called SDS. Proteins are denatured and given a uniform negative charge before being loaded onto a gel. Electrophoresis is then used to separate the proteins based on their molecular weight. SDS PAGE is commonly used for protein analysis and quantification. Native gel electrophoresis is a technique that allows for the separation of proteins in their native, folded state. Unlike denaturing techniques such as SDS PAGE, native gel electrophoresis preserves the native conformation and functional properties of proteins. The principle behind this method is to separate proteins based on their size, shape, and net charge. During electrophoresis, an electric field is applied, causing proteins to migrate through the gel based on their charge to mass ratio. Native gel electrophoresis is commonly used to study protein complexes, enzyme activity, and protein-protein interactions. Two-dimensional gel electrophoresis, 2DGE, is a technique that separates proteins based on their isoelectric points and molecular weights. This high-resolution method allows for the simultaneous analysis of numerous proteins and is commonly used in proteomics to identify biomarkers in complex protein samples. In Western blotting, proteins are separated using gel electrophoresis and transferred onto a solid membrane where they are immobilized. The membrane is then probed with antibodies specific to the protein of interest, allowing for the detection and analysis of specific proteins within a complex mixture. This technique enables researchers to study protein expression levels, post-translational modifications, and protein-protein interactions. Capillary electrophoresis, CE, is a technique that separates charged molecules like proteins and nucleic acids in narrow capillaries filled with an electrolyte solution. It offers better resolution, shorter analysis times, and lower sample consumption compared to gel electrophoresis, making it useful in genomics, proteomics, and pharmaceutical research. Isoelectric focusing, IEF, is a technique that separates proteins based on their isoelectric points, pi, a pH at which proteins have no net charge. In EF, a pH gradient is created along the gel matrix by employing chemicals termed ampholytes or immobilized pH gradient, IPG, strips. When an electric field is applied, proteins migrate through the gel until they reach a region where the pH matches their pi, resulting in a sharp band. EF is widely used to investigate post-translational modifications, assess protein purity, and separate proteins with similar molecular weights but different pi values. DNA sequencing is essential in molecular biology to determine the sequence of nucleotides in DNA. Capillary electrophoresis, CE, is vital in modern sequencing methods. During CE, fluorescently labeled DNA fragments are separated by size and detected by analyzing fluorescent signals. This helps determine the DNA sequence. Pulse field gel electrophoresis, PFGE, is a powerful technique used for the separation of large DNA fragments, particularly in the analysis of genomic DNA. Traditional agarose gel electrophoresis is limited in its ability to separate large DNA fragments, but PFGE overcomes this limitation. In PFGE, an alternating electric field is applied to the gel, changing its direction periodically. This prevents the formation of entangled DNA structures and allows for the separation of larger fragments. PFGE is commonly used in molecular biology research, including the study of genomic rearrangements, identification of bacterial strains, and mapping of chromosomal DNA. Denaturing gradient gel electrophoresis, DGGE, is a specialized technique that separates and analyzes DNA fragments based on their sequence variations. DGGE takes advantage of the fact that DNA molecules with different base compositions denature at different temperatures. In this technique, a linear or nonlinear denaturing gradient is created within the gel, which changes in chemical composition or temperature. When DNA samples are electrophoresed through the gel, the fragments with similar sequences denature at specific positions along the gradient and form distinguishable bands. DGGE is commonly used in mutation analysis, microbial ecology, and genetic research. Single-strand conformation polymorphism, SSCP, analysis is another DNA analysis technique that exploits sequence variations in DNA fragments. Similar to DGGE, SSCP relies on the different conformations assumed by single-stranded DNA fragments with different sequences. In SSCP, DNA fragments are denatured, and single-stranded DNA molecules are separated based on their conformational differences. The separated single strands are then visualized as bands on a gel or detected using fluorescent methods. SSCP analysis is used in DNA mutation detection, genetic linkage analysis, and biodiversity studies. Gel electrophoresis techniques revolutionize cell biology by separating, analyzing, and characterizing biomolecules. From simple agarose gels to high-resolution capillary electrophoresis, each method offers unique advantages for studying cellular processes, diagnostics, drug development, and disease mechanisms. Their importance in cell biology research will persist with evolving technology.